Hi, everyone, and welcome to today's Hot Topic program. How do paleoanthropologists identify our evolutionary relatives with Bernard Wood? My name is Briana Pobiner, and I'm a paleoanthropologist and educator at the Smithsonian's National Museum of Natural History. I'm a brown and gray haired woman in a gray shirt with a purple scarf wearing headphones in front of a Zoom screen with an African savanna photo with grass and an acacia tree behind me. Whether this is your first time joining us for the Hot Topics or you've attended before, we're so glad to have you here. Before we get started, I have a few housekeeping notes. This discussion offers closed captioning. You can turn the captions on or off via the CC button, which should be located at the bottom of your Zoom interface. We are in a webinar format. We can't see or hear you. So as you have questions, please go ahead and submit them to the Q&A box, which is at the top or bottom of your screen, so we can sort through as many questions as possible. The Q&A box is also where we will share any relevant links during the program, so keep an eye out there. We'll start with a presentation by our speaker, Dr. Bernard Wood, and then I'll join him here to take your questions. And during the presentation, I will also write answers to some of your questions, at least any that I can answer, as will another member of our behind-the-scenes team, Dr. Ryan McCrae. So now I'd like to go ahead and introduce our speaker for today. Dr. Bernard Wood is University Professor of Human Origins at the George Washington University. He studies hominin systematics, comparative anatomy, and soft tissue-based phylogeny in primates, with interest in improving species recognition, phylogenetic reconstruction, and distinguishing reliable phylogenetic signals from noise in the fossil record. He has studied many original hominin fossils and authored or co-authored over 250 scientific articles and 20 books. In 2023, he received the Royal Anthropological Institute's President's Lifetime Achievement Award in recognition of his over 50 years as a leading figure in human origins research, his focus on hominin adaptations and evolution, and his mentorship of numerous PhD students and postdoctoral researchers. And now I'd like to invite Bernard to share his presentation with us.
Many thanks, Briana. Can you hear me?
Yes, I can hear you great. We can see you great.
Okay, and you can see the slide?
Not yet.
Okay. Okay. How about that?
Perfect.
Okay.
All right, take it away.
Okay. Okay, well, hello, everybody, wherever you are. Thank you for joining us. I'm going to just run through some images that I still call slides, even though they're not in a projector, and I will get going. This is a picture of Charles Darwin, and the reason I'm showing you the picture is because of Darwin's contribution, which includes the notion that all living things are part of a single tree of life. So this is an oil painting of him at the college where he was in Cambridge. And the next is this is an early photograph. It's not long after Henry Fox Talbot invented photography. And it's a photograph of Darwin the year that he died. So this is the metaphor that he introduced. This is a tree. You can imagine this is three-dimensional and not the two-dimensional tree you see here. You can see it has a trunk, and so there is a common ancestor. And all the species alive today are on the surface of the tree of life, if you can imagine that. And all the animals that have ever been alive in the past, they are within the tree of life. And we are at the end of just one of the twigs which runs towards the surface of the tree. And my task today is to try and explain to you how we identify the adjacent twigs, the ones that are closest to us. the term I've already used is species, and it's part of the way that we describe the world. Now, rather than dwell on the species, let me give you a couple of examples of taxonomies of which a species is a part. It's the taxonomy, how we describe living things and how we describe living things in the past. So let me give you an example of a taxonomy. So we have an inclusive category. Let's say it's a powered vehicle. It could be a spaceship, or it could be a commercial airliner, or it could be an Amazon delivery truck. And then automobiles are a subcategory. Rolls-Royce automobiles are a subcategory of automobiles. The silver shadow model of Rolls-Royce is a subcategory of Rolls-Royce. And the model that was begun in 1970 that's called the Silver Shadow 2 would be the equivalent of a species if we were applying it to cars. If you're not a car person, how about saying something like fine arts? Then paintings are a subcategory of fine arts. Oil paintings are a subcategory of paintings. 20th century oil paintings are a subcategory of oil paintings, and Picasso's paintings, and then Picasso's anti-war paintings would be the lowest, the least inclusive, the most exclusive category. And that is one of them, but you can't classify a single painting. You put it into the species equivalent, which is Picasso's anti-war paintings. So now I've confused you with trying to give you metaphors. Let me explain what I'm trying to do in this talk. So I'm trying to answer two questions. The first is, who are our own species' closest living relatives, and when do we share a recent common ancestor with them? And the second question, does our twig of the tree of life consist of just the ancestors of modern humans, in other words, our ancestors, or do we also have close fossil relatives that did not make it onto the surface of the tree of life? So those are the two questions. The first time I became aware of human evolution was when I took a course in human evolution in 1966. It's a long time ago. I worked for 12 years in Liverpool, so I'm expressing that in terms of the Beatles. And 1966 was when the LP came out, The Yellow Submarine, and you can see the songs that are in it. So it's a long time ago. When I was taught, the conventional wisdom was that hominids, which we were the sole living example, were separate from pongids, which included the African apes and the orangutans. And so the distinction between us and our closest living relatives was made at the level of the family. And if we go back to our analogy, that I think would be something like oil paintings. And in 1966, there wasn't much known about the fossil record of the animals that were thought to be closely related to us. And I'm going to show you a series of diagrams, and the blue ones, they are early hominins that if you saw them on the metro in DC, you might be slightly disturbed, but I don't think you would go to another carriage at the next stop. Homo habilis, you probably might go to the next carriage at the next stop. the next ones, Paranthropus robustus and Paranthropus boisei, that were known in 1966, and Australopithecus africanus, you would very definitely move to the next carriage at the next stop, because these are would have been really not terribly like us, maybe Australopithecus africanus a bit, but the Paranthropus robustus and Paranthropus boisei, they would not look at all like modern humans. So now what's the situation? So I've told you what the situation was in 1966. Now we separate modern humans from the African apes and orangutans at the level of the tribe. And so we are in the hominini, and so we use the informal term hominins. The chimpanzees and bonobos are in the panini, and so we use the informal term panins. We have a lot better information, but still not very good information about how they are related to each other. And based on the genetic differences between modern humans and the living African apes, the hypothesis is that we probably shared a common ancestor with chimpanzees and bonobos about 8 million years ago. Now, if this upsets you, just imagine how upsetting it must be for chimpanzees and bonobos and gorillas, because they are in the zoo, and the people that come to the zoo and buy ice creams and look at them, they are more closely related to one of them, to the chimpanzees and the bonobos, than they are to the gorillas. So I think it will be a lot more upsetting for them than it is for us. So, so a tribe would be, something equivalent to the 20th century oil paintings. So my research interest is trying to work out what happens between the hypothetical common ancestor around 8 million years ago and modern humans. And that's what I want to talk about today. What I need to explain is that the evidence that I'm going to be talking about comes mainly from Africa. But Africa is a big place. And the evidence I'm going to be talking about comes from the East African Rift Valley, which is the red balloons on the right. It comes from a series of caves in southern Africa at the bottom of the picture, and there is one site which is not in either of those places. So when we talk about the fossil record, it has to be with the caveat that we are looking at the fossil record which has been recovered from these two places. There clearly must be a lot of other evidence, but we don't have access to it. And the other thing I need to explain, and this is Ryan is going to be helping with the questions. On the top left, there is a pie chart which shows the relative numbers of items of information from a complete modern human skeleton. And you can see, the axial skeleton, that will be all the vertebrae and the sacrum and so on. You can see the teeth and so on. If you look at the pie chart on the right, the large pie chart on the right, that is what the hominin fossil record looks like. So we are reconstructing the hominin fossil record on the basis of mainly information about the teeth, with very limited information about the limbs and the axial skeleton. So you need to take that into account when you are trying to understand what I'm trying to explain to you. So the fossil record comes from limited locations geographically, and it is and the interpretations are based on a very odd sort of individual that would be all teeth and hardly any limb bones and axial skeleton. So how do we reduce our ignorance about human evolution? You find more evidence, or you think of smarter ways to look at the existing evidence. And that's what we have been doing in the past few years. For example, Matt Skinner now works in Leipzig at the Max Planck Institute, but he was a graduate student a long time ago. And you can see on the left of the image, you can see an occlusal view of some teeth. And we thought we were very, very scientific when we measured a tooth. We measured its length from the front to back, and we measured its width in two places. We thought that was amazingly sophisticated. And then we gathered information about the shape of the occlusal outline of the crown. And then we got even more sophisticated, and we divided the crown up into the major cusps, and we recorded their areas. So we thought that was incredibly sophisticated. Now people use computer tomography. They use three-dimensional geometric morphometrics to collect a lot more information. And you can see an example on the right. So that's an example of getting, looking at the existing evidence in a sort of smarter way. So what are the research questions? How many species are there in the human fossil record? Well, this is what a splitter like me, in other words, someone who would have a tendency to recognize more species rather than fewer species, this is what it looks like in 2026. If you remember how few species there were in the 1966 version, you can see that all the attempts to collect fossils in the intervening period have been successful. These may or may not be species that will survive the test of time, but nonetheless, this is a sort of speciose interpretation of the hominin fossil record. On the left is millions of years, and so each column is a hypothesis about the existence of that species and its first appearance date at the bottom and its last appearance date at the top. So why might there be even more species than are in that sort of speciose interpretation of the hominin fossil record? Well, one reason is the spatial scope of the hominin fossil record, and I've already talked about that. We only have information from a relatively limited part of Africa. There may be hominins that were living in other parts of Africa that we don't have access to. The hominins could be living in a part of Africa, but if they were fossilized, but if we don't have access to the strata in which they were fossilized, then we just don't find them. It doesn't mean they're not there. It's just we're not going to have access to them. We should also realize that the hard tissue evidence that forms the basis of the hominin fossil record may or may not be the reason why primates mate with each other and don't mate with other primates. This is called the specific mate recognition system. So, for example, guanons are monkeys. They mate with each other according to the color of the facial hair. And so green-faced guanons mate with green-faced guanons. They don't mate with blue-faced guanons. And the other reason that there may be more species is that hominins are a very rare part of the mammalian fossil record. They make up about 1 or 2% on average of the mammal fossils found at any site. So you might have a site, you might have quite a lot of bovid fossils or quite a lot of pig fossils, but you may not have hominins. It's just because you haven't been looking long enough and you haven't, they may be there. It's just you can't assume they're not there. So why might there be fewer species? Well, if you just think about this, the thought experiment. I'm a pretty simple guy, so this is not a very sophisticated image. And it took me a long time to make all these gray circles. But just imagine that there is a species and it's gray circles. Okay. The average length of a mammal species in time is somewhere between 1 or 2 million years. So just imagine the vertical axis is a couple of million years in time. Let's imagine this hominin species was spread over, say, a couple of thousand kilometers. That was its range. So this is a hypothetical species. You and I know they all belong to the same species, but they are not identical because that's the nature of a species. In some parts, they're small. In some parts, the grayness is a little more exaggerated, so they're dark gray. In some parts, they're light gray. Believe me, they all belong to the same species, even though they're not identical. And that is a lesson that some of my colleagues really never learn, that a species has its own variation, and that variation is dictated by climate and environment. In some parts of the range, the individuals will be larger than others because there is more food. In other parts of the range, they will be smaller because there is less food. In some parts of the range, they would have larger chewing teeth, and in other parts of the range, their chewing teeth would be smaller. But this is the same species. So somebody gets some money to go to Africa to look for hominin fossils, and they find a site, and that site maybe samples 300,000 years, and they discover a hominin, and they get themselves a paper in Nature. So that's fine. You and I know that this other site, it's sampling the same species. Because it's the same species, even though the information is really valuable, the researchers try and submit their paper to Nature or Science, but it's rejected, and they end up publishing it in the Journal of Human Evolution. Then somebody gets some money and they go to a different part of Africa, and they think, wow, we found some stuff that doesn't look like the large light gray circles. The circles are smaller and they're slightly darker. Wow, we can write a paper for Science or Nature saying we found a new species, but you and I know that it's the same species. So that's why one needs to think that some of the more speciose interpretations may not be correct, although I'm inclined to think that they will eventually be shown to be more correct. But nonetheless, if we express the hominin fossil record in the smallest number of species that we can use to interpret it, it still looks like this. And it's still the case that from just more than 4 million years ago until the recent past, until the last Neanderthals or Denisovans died out, there has always been more than one hominin existing in the world. So how are the species related to one another? Now, if there's more than one hominin existing in the world at the same time, they can't both be our ancestors. And so we are, the problem here is to sort ancestors from close relatives. So the question is, does our twig of the tree of life consist of just ancestors, or do we also have non-ancestral close relatives? This is a picture of my father's family, and it was a golden wedding anniversary, and the picture was taken in 1933. All the people in the picture are my relatives. On the left is my father's father. If you go up a bit and look at the A, that's my father. If you go to his left, but our right, that's his mother. If you go down, that's her mother. And if you go to the right, the other side of the rather nice table decoration, that's her father. The R is Aunt Kitty. She was delightful, but she was not my ancestor. She was a close relative. Okay. Aunt Kitty was a close relative and not an ancestor. So in that picture, the ratio of ancestors to close relatives is high in favor of ancestors. This is my christening. And you can see that there's a mixture of relatives and family friends and godparents. And the relatives are identified there. You can see, well, you don't have to be a genius to realize who I am there. However, in this picture, the ratio of ancestors to close relatives is actually there are more close relatives than there are ancestors. The only ancestors of mine in that picture are my father's father on the left. My mother is the next A. My father was still away in the war at the time. And then my father's mother is the next A. And then to the right was her mother. So there are 4 ancestors, but there are a larger number of close relatives. So is the hominin fossil record more like the Golden Wedding or more like my christening? It's probably more like my christening. In other words, I think there are a whole bunch of close relatives in the hominin fossil record that aren't necessarily our ancestors. So just to remind you of the rather specious interpretation of the hominin fossil record, most of those will be close relatives and not ancestors. And even in the lumpers interpretation, the balance would still be most of those will be close relatives and not ancestors. So in theory, reconstructing relationships should be simple, because the more morphology you share, the more closely related you are. But there is a bit of a sort of but I wish life was that simple, but it's not. And this is a picture of the Piltdown Committee looking at the Piltdown discovery. And the guy who you can hardly make out on the right is the person who I'm going to focus on. And he was a guy called Sir Ray Lancaster. He was the director of the Natural History Museum in London. Apparently, he was a perfectly awful manager, but he was interested in relationships, and he wrote in 1870, when identical or nearly similar forces or environments act on 2 or more parts of an organism, the resulting correspondence is called forth in the several parts of the 2 organisms. This is very flowery language, it's very Victorian, will be nearly or exactly alike. So he's really saying that you can get very similar sorts of animals because they are responding to the environment, not because they are necessarily closely related. And he says, I propose to call this kind of agreement homoplasis or homoplasy. Neither of them really caught on as an idea. Eventually, his idea was called homoplasy. And so homoplasy refers to features shared by two taxa that were not present in their most recent common ancestor. Such morphology gives the false impression that the two taxa are more closely related than they really are, and it complicates attempts to reconstruct phylogenetic relationships. And we know that's the case. We know that homoplasy is rife in groups like cetaceans, which have a much, much better fossil record than hominins. And so it would be crazy to imagine that hominins were not affected by homoplasy, which means that trying to recover relationships is a rather harder problem than we would like it to be. And so it remains, it really remains a significant challenge. So if we go back to this sort of speciose interpretation, and we look at the green ones down in the bottom right, these are all taxa that their discoverers were convinced were the earliest hominins. Now, they can't all be the earliest hominins. And so what are the options for creatures that don't look like modern humans? They don't look like chimpanzees or bonobos. They're more chimp-like than modern human-like, but they have some things that chimps don't have. They have some things that suggest that they had a slightly different locomotion than modern chimps, and that their diet was probably less reliant on fruit and so on. And so where might these creatures be accommodated in that branching diagram I showed you earlier? Well, they could be our close relatives. Okay, they could be our close relatives, or they could be on the main red line to modern humans. In other words, they could be our ancestors. But how you tell ancestors from close relatives is a struggle. They could be closely related to chimpanzees and bonobos, or they could belong to a group for which there is no living representative, which is actually closer to modern humans than modern humans are to panins and bonobos. So, having confused you and tried to persuade you that what on the surface might look a simple problem is not that simple, just a few consumer warnings. Where the fossil evidence is found is not necessarily where the action was. In other words, a lot of interesting evolutionary history, hominin evolutionary history, may have been going on in places where we have no fossil sites. Those first appearance dates and last appearance dates, they're really complex phenomena that are not really conducive to simple explanations. So, you want to avoid, saying, well, the climate changed, so therefore these things evolved. It's probably rather more complicated than that. And another consumer warning is that events are what processes look like when they are sampled discontinuously. And so it's easy to see, oh, something happened, but you just don't have access to what happened before. And I'm a great fan of Albert Szent-Györgyi, who said, simple solutions to complex biological problems are almost certainly wrong. So before I finish, I just wanted to explain how indebted I am to the people who find the fossils, because without the fossil evidence, I wouldn't have had a career. And these are the Kenyans that found most of the fossils from Kubifora that was on the eastern side of Lake Turkana, where I worked. And I want to express my thanks to Richard Leakey, who was told by his mother in 1968 that he should take this young medical student with him to northern Kenya, and um, and depending on Richard, how frank Richard was being, he sometimes thought that was a good thing, and sometimes thought that maybe, especially when my interpretation of the hominin fossil record differed from his. And I want to thank you all for listening.
Wonderful, thank you. I particularly enjoyed that picture of some of the hominin fossil hunters with the Land Rover that looked like it was sort of cut off halfway up. That was great. Wonderful. So we have a couple of questions. I'm going to actually start with some behind the scenes questions. So I'm gonna ask the first one, which is considering the numerous ways that scientists can identify and separate species. So these are often called species concepts. Are there any that are particularly useful or not useful, do you think, for the hominin fossil record? Or is trying to, follow any number of species concepts ultimately not useful at all?
Well, I think you have to have in mind the species concept that you think intellectually it makes most sense, but then you have to think uh, that the hominin fossil record is made up of hard tissue evidence. And
Hmm.
if you think of the guanan example, the facial color of the guanan would not, I mean, there is no fossil record of that.
Hmm.
And the problem is that if you look at skeletons of guanans, they are almost indistinguishable.
Hmm.
So you would never, you know, if you just looked at their skeletons, you would say there was one species of guanan. But if you go out into the wild, or if you go to a zoo that specializes in guanans, you will realize that that would vastly underestimate the number of species. So,
Hmm.
so the soft tissues don't preserve, and the soft tissues are probably where most of the species of the signals that animals use to decide who to mate with. It's mainly soft tissues.
Yeah, that's a good point. Thank you. Um, so speaking of things that do preserve, talking about genetic evidence, here's another behind the scenes question. Considering well, sometimes genetic evidence preserves when we're lucky. So considering the genetic evidence of later hominins like Neanderthals and Denisovans interbreeding, and the paper that just came out recently that might include Homo erectus interbreeding with Denisovans, how does this or does it complicate our picture of how species are separated? So that's the first part of the question. Then I'll ask a second part after.
Well, the if you think of living baboons, um,
Hmm.
uh, baboons in Africa extend all the way from North Africa down to Southern Africa. If you look at the baboons in Northern Africa, they are they're obviously distinct from the baboons in Southern Africa. But if you look at the baboons in Ethiopia, which are in another species, they interbreed with the adjacent species. Now, the interbreeding is not so much that it blurs the distinction. It just means that at the boundaries, you will find individuals that have some characteristics belonging to one species and some characteristics belonging to another. But it doesn't mean that the baboon species aren't a reality.
Hmm.
So so even if hominins did interbreed, which it would be crazy to think that they didn't.
Hmm.
The question, my response will be that the extent of interbreeding did not blur the distinction that we see in the hard tissues in the fossil record.
Hmm. That makes sense. And actually, this you may have already answered the second part of this question, maybe, but so given this interbreeding, which I think we would expect, do you think that this interbreeding was present all throughout our evolutionary tree, or is it really a more modern hominin phenomenon with these like very closely related taxa like humans, Neanderthals, Denisovans?
My sense is that it would have been, there would be more of it recently.
Mm-hmm.
You should understand that, as soon as our ancestors looked at all like modern humans, I lose interest. And so, it's wonderful to read these papers about sort of Denisovans and Neanderthals. Um, but I'm sort of more interested in creatures that really look sort of different
Mm-hmm.
and so different. If you take Paranthropus boisei, it's such a weird creature.
Mm-hmm.
There's no living primate that makes a reasonable model for trying to understand how this creature made it, how it lived. And so I'm sort of more interested in the relatives that come to your Thanksgiving dinner that, you're not quite sure about, but somebody tells you that you're related to them, so they should come to the Thanksgiving dinner. So I'm more interested in those or their
Um,
equivalent.
so actually, this leads on to another behind the scenes question. Maybe you just answered it. Do you have a favorite species of hominin or a favorite fossil? And if so, why is that your favorite?
Um, well, my favorite fossil is the type specimen of Paranthropus boisei, and it's called Oldobai hominid 5. And when I was doing my research, it was actually in a museum in Dar es Salaam. And Leslie Ayala and I went to see the material there. We clearly didn't have to make an appointment because we we think that nobody had been there for 5 years before we went there. The safe in which the skull was in a cupboard. There was no light bulb in the room, and there was no light bulb in the cupboard. So we had to go out to a store and buy some light bulbs. And so we eventually got OH5 out of the safe. It is the most fantastic specimen you will ever see in your life. It's just amazing because it's so perfectly preserved. Much of it's perfectly preserved, but it's so weird.
Hmm.
So the answer to that would be OH5 on Paranthropus boisei.
All right, thank you. That's good to know. And if I'm going to get you a thank you gift for doing this talk, it might have an OH5 theme. So I'm going to ask a question that was sort of going off one of our audience questions. The initial question was about why are Denisovans not on your initial species list? And the answer was, well, they hadn't been found yet
Yes.
in that 1966 list. But
Yes.
to sort of go off of that, are there any potential species of hominins that you don't include on your chart that you think might be species, but like are maybe less well accepted?
No, I think the chart includes um, all the runners and riders, I think. Uh,
Okay.
Even the ones that, aren't going to, they aren't going to be placed in the race, but nonetheless, you
Okay.
should have them on the board.
Got it. Um, so I might ask you a few sort of career related questions. One is, so I know you started your career in medicine, you were dean of a medical school. How did you get into paleoanthropology?
Well, I got into paleoanthropology because when I was a medical student, I did an extra degree in anatomy, and I did a course on human evolution. And the
Mhm.
course on human evolution was taught by a guy called Michael Day, and he had worked on the fossils discovered by Lewis and Mary Leakey in Olduvai Gorge. Um, Michael Day's wife, you did ask me. Michael Day's wife was a primary care physician. She used to look after Mary Leakey, and they were very good friends. And so, you know, somehow, sort of Mary said, well, I think this, young man who's, a student should go up to northern Kenya. I'll have a word with Richard. And so basically she went home and said, Richard, I think you should invite this guy. So so I went. I I I enjoyed it. Richard tolerated me. I mean, you've been in the field, you know perfectly well, that the people you want to be in the field with are not people who drive you crazy,
Yes.
you, and so I had enough, I had enough sort of wisdom to make sure that I wasn't a nuisance, and we got on well together, and he invited me back, and he invited me to describe the fossils that he was finding along with other people. And so I did surgery. And I had to go back to do the exams for the fellowship of the Royal College of Surgeons. And I kept putting off doing that exam because I kept doing more research. And eventually eventually I became an examiner for the anatomy exam for the Royal College of Surgeons. And I thought, I'm not going to, now I'm an examiner, I just don't have the chutzpah to take the exam, because supposing I failed it. And so basically, so I just sort of left medicine. I mean, I worked as a surgeon for a decade on and off, I did surgery part-time.
Okay.
But then I eventually stopped.
Okay. I'm going to turn to an audience question. And so Walter asks, how much detail of the surrounding past geological environment is studied and correlated with the description of a species? Could this contribute to homoplasy?
Yes. Yeah. I mean, I think it's what sort of drives homoplasy, which is why we are so keen to learn from our colleagues who are expert in interpreting the sediments, interpreting the other fossils, to try and paint a picture of the habitat that the early hominins were living in. So I think that's a very astute question.
Okay. And let me I'm just looking at some more behind the scenes questions. So can you, to follow up on this one, can you elaborate on homoplasy? Are there particularly important examples in the fossil record, or good illustrative examples?
The problem is that in the fossil record, you don't have, the reason that we know homoplasy exists is because people use molecules and latterly the genetic information about DNA for working out how animals are related.
Mm-hmm.
So we have information about relationships which is independent of morphology.
Yeah.
The problem in the fossil record is that we have that, for Denisovans and for Neanderthals, but there is really no sort of good DNA information much older than about 500,000 years. And so the problem is we don't have a non-fossil evidence related line of evidence about relationships.
Yep.
And so we just have to do our best. But I think, you need to look around you and see, well, is there homoplasy? Yes, there is. And why do you think hominins should be somehow exempt from homoplasy?
Right.
And I just think it's just crazy to make.
Yeah. I'm going to turn to another audience question. So Stephen asks, will the new science of protein analysis, as opposed to DNA, help in distinguishing species, do you think?
Yes, it is. And it's sort of almost counter, intuitive. You would have thought that sort of proteins would be easier to get at than DNA, but it turns out that the proteins. So, for example, there is a difference in one of the proteins in our teeth. It's a protein called amelogenin. And there is a sex difference. So that helps us identify fossils as males and females. So, for example, Paranthropus boisei, one of the things that interests me about it is that the males are not just larger versions of the females, there are shape differences between the males
Mm,
and the females. And so if we have non-morphology related ways of telling males and females, it allows us to say that is what we call within species or intra-specific variation. It's
mm-hmm,
not the difference between two species. Now,
mm-hmm,
there could be a male of one species and a female of another species, but if your working hypothesis is that this is one species,
mm-hmm.
and I think these are the large males and these are the small females in the species, and that is confirmed by the amelogenin, then that,
Mm-hmm.
makes you feel good.
Exactly. All right, I'm going to go to another audience question. Tad says, great talk. I love the family photos as a teaching tool. So in this talk and others, I hear a lot about our common ancestors with other living primates, and Dr. Wood identifies that at about 8 million years. But has anyone tried to trace our ancestry back earlier than that, like all the way back to when the asteroid hit 65 million years ago and mammals first began to emerge?
Yes, but it's a different scale of problem.
Mm,
I mean,
yeah,
if you're in research, I mean, it's like going on safari. You know, you wake up in the safari lodge and you take your coffee outside and you see elephants and giraffe and you see baboons and you see everything. If you're a researcher, you need to choose which of these animals you're going to focus on.
mm-hmm,
You can't look at elephants for 10 minutes and then start to do some research on these. And so basically, each of us really has to decide. I have some colleagues who are interested in the origin of mammals.
mm-hmm,
I have colleagues who are interested in the origin of primates.
mm-hmm,
I have colleagues who are interested in the origin of apes,
mm-hmm,
but that's not my bag, you know.
yeah, mm-hmm,
And so I think you have to understand that if you do research, you have to pick where you think you can reduce ignorance.
mm-hmm.
And it's really difficult to do that across, a whole range of research questions. I'm
Yeah,
not sure
that makes
whether that answers the question.
I think so. Yeah. I mean, I think the answer is yes, and that there are people who ask these questions sort of particular places in a sense
Yeah.
on
Yes,
the our big
yes,
family tree. Yeah.
yes. I mean, if you look at the
Yeah.
tree of life,
Yeah.
there are researchers interested in all sorts of, in all sorts of places on that tree. And you have to
Yeah.
realize, if that was literally the tree of life, the vast majority of that tree would be bacteria and would be,
Yeah.
you know, uh, uh,
Mm-hmm.
would be organisms without backbones and things like that. And,
Mm-hmm.
and so I'm only interested in a tiny part of that tree.
Yeah, exactly. All right, another audience question. John asks, and this may be a bit outside of the fossil record, but lacking evidence of gene flow due to the age of samples, is there any degree to which we can see flow or sharing of lithic or other technologies to indicate relationships between populations? He says, or are the sample sizes too small?
That's a very interesting question and a very sensible question because
Mm-hmm.
lithic technology is lasting evidence of behavior.
Mm-hmm.
And you could say, well, this hominin made stone tools this way,
Mm-hmm.
this hominin made stone tools that way, and if you could link those. The problem is that, there are only a few ways that a stone can fracture.
Yeah.
So basically, you have the homoplazy problem coming in
Mm-hmm.
that, if you bash one stone against another, then, just a limited number of things could happen.
Mm-hmm.
And we have been working, and in fact, we've written about this pretty recently, and it's a struggle to link stone tools with hominins.
Yeah.
And you have to realize that hominins were probably doing a hell of a lot else other than making stone tools. It's just that we tend to, because that is lasting evidence of behavior, that's what
Hmm.
we focus on because it's there.
Exactly.
But we need to remember that there may be lots of behaviors that they were indulging in that leave no lasting evidence.
And I agree that sort of assuming that a stone tool technology was only made by one hominin species is, I think, an assumption we should question. So,
Yes,
yeah,
yes, yes. It's certainly not where I would sort of start, I think.
yeah. Here's another audience question from Misha who asks, perhaps this is an oversimplification based on teaching children, but when I was in grammar school, we were taught that species are species because they do not breed with members who are not of their species.
That's right.
Given that modern humans, as I understand, have both Neanderthal and Denisovan DNA, how useful is such a fine distinction as species?
Well, I think it's still useful.
Hmm.
I mean, the fact that it may not be the definition that we were brought up with in a biology textbook, it still means, that let me put it this way, if it wasn't useful, then everything would blur together.
Yeah, yeah.
And it doesn't, you know,
Hmm.
it doesn't.
Hmm.
Most of the hominin fossil record, Paranthropus boisei is very distinctive. It's really difficult to confuse it with early Homo.
Hmm.
However, there are some specimens, that have a bit of this and a bit of that, but
Hmm.
mostly it's very distinctive.
Yeah.
And so I think interbreeding, I think is a reality, but it doesn't blur the major distinction that is preserved.
Yeah. That makes sense to me. All right, I think we have time for one or two more questions, and I'm going to privilege a behind the scenes question. So, this question is the number of species, the number of fossils, the general understanding of human evolution has increased through time and for sure over the course of your career. Do you see this increase being linear, exponential? Is it tapering off? How would you predict that pattern to continue into the future, especially with new techniques like ancient DNA and proteomics?
That's an interesting question. I mean, I think there must come a point when we are not going to collect any more information which is going to change, an interpretation very much.
Hmm.
Um, my experience is that when you only had a few species, everything looked pretty obvious.
Hmm.
There was a time when, you had rather more information and things look, rather confusing.
Hmm.
And you collect a little more information and some of the confusion gets answered.
Hmm.
My sense is that you have to remember what your research questions are,
Hmm.
not get sort of deflected by the latest sort of shiny object. And
Yeah.
and so that's my reflection from doing this rather longer than probably anybody with any sense should have done it.
Well, I think we've enjoyed the perspective from the course of your career, and I will ask the last question. Also, this question is actually for me. So, kind of following on the question we just asked, what is the discovery over the course of your career that was most surprising to you?
Most surprising. Well, my discovery or other people's?
Anybody's discovery,
Oh,
or
um,
your discovery. I say
no, no,
interpret
no,
it as you wish.
no, no. Um, you know, I'm not sure there was anything that was most surprising. Sort of something like Sahelanthropus. You know,
Hmm.
I wouldn't have necessarily um, predicted all the details, but you sort of in your head knew
Hmm.
that there were creatures likely to be creatures like that.
Hmm.
I'm sorry, I'm not, I guess I'm not being very, there's nothing sort of terribly surprising. And to be honest, if you're a half decent scientist, you shouldn't be surprised.
Well, I was going to say, I think the fact that that nothing springs to mind just means that you've been thinking really broadly about this and that there hasn't been anything so left field that it
Sure,
didn't really fit in our understanding in general.
sure. Yeah,
Yeah.
there are a few things, where people say, well, you know, Sahelanthropus isn't a hominin, it's a fossil gorilla, and you sort of, you think, well, what were you drinking? It's not a fossil gorilla because it doesn't look anything like gorillas. It just,
Hmm.
if you looked at it from the other side of the room, it might look like a gorilla, but if you look at a car at the other side of a large car park, it's going to have an engine compartment, a passenger compartment, and a trunk. Well,
Hmm.
that's, most cars look like that unless they're SUVs, and that's not terribly helpful. So, yeah, yeah.
Yeah, that's great. Thank you. All right, so I will wrap up. So we'll conclude today's virtual program. Please join me in thanking Bernard for sharing his work and his thoughts with us. I'd also like to give special thanks to those who made this program possible, to our behind the scenes team who helped sort through your questions, including Dr. Ryan McRae, who works with us in the Human Origins program, to our donors, volunteers, and viewers like you, and finally, to all our partners who help us reach, educate, and empower millions of people around the world today and every day. We thank you. This was our last hot topic of the spring season. We'll be back in the fall starting on September 24th. So keep an eye on your email for updates about the fall season of Hot Topics. We've put a link in the Q&A where you can find information about our other upcoming programs and how to sign up for the museum's weekly e-newsletter. That's the best way to stay informed about upcoming programs, Hot Topics, and others, and to learn more about the museum's research and exhibits. After this webinar ends, you'll see a survey pop up asking for some feedback about the program. Please take a moment to respond. We're very curious to know what topics you might be interested in seeing for future programs, and we appreciate your input. Again, thank you to Bernard, thank you to the audience, and we will see you in the fall.